All right, welcome everybody to lecture number 3, amino acids. So for today's lecture, we'll start out with a very quick review of chemical functional groups. So this should be review from your organic chemistry courses, but in case you forget, we'll go over them very quickly just so you know, because they are important to understand the different flavors of the different amino acids. Then we'll go into an introduction to amino acids. So amino acids are the building blocks of proteins. And we'll talk about the chemical characteristics that each of the amino acids share. Then we'll go into descriptions and categories of the 20 different amino acids that build proteins. And I'll go through this for the majority of the lecture because I want you to remember each of the 20 amino acids. So their chemical structure, their full name, their shortened name, and their one-letter codes. So I know this seems daunting, but it's going to be to pay off in the long run. You'll be able to answer a lot of exam questions if you know all 20 amino acids. So you're going to have to bust out those flashcards or however you want to learn them, but you should learn all 20 amino acids. And then we'll end the lecture with just a quick discussion on what is an essential versus a non-essential amino acid. So let's launch into it. Lecture number 3, amino acids. So for our lecture today, I want to start out with a quick discussion of functional groups. So I'm just going to review this information. You should have already discussed it in other chemistry classes, but it is going to be important when we're talking about the different types of amino acids. So the first functional group we'll talk about are hydrophobic functional groups. And so one class of compound that would contain a hydrophobic group are hydrocarbon chains, so aliphatic hydrocarbon chains. And so they would just have these hydrocarbons, so CH3, so these are going to be hydrophobic. And an example of an aliphatic hydrocarbon chain would be in the amino acid alanine, and it is shown right here. You can also have aromatic hydrophobic groups. So these are hydrocarbons in a ring structure with multiple double bonds. You can see an example of this right here. And then an example of an amino acid that has this functional group would be phenylalanine. You can see it has this phenyl ring right here. Another functional group are hydroxyl groups. And so a class of compound that contains this functional group would be alcohols. And so there's this OH group right here. So that would be referred to as a hydroxyl group. And an example would be this group here shown on the molecule ethanol. So next we'll talk about aldehydes. So these don't come into play with amino acids, but they do when we're going to talk about carbohydrates. So aldehydes, so the class of compounds that contain aldehyde functional groups are just aldehydes. And so they have this carbon double bonded to an oxygen and also bonded to a hydrogen atom. And an example of this would be acetaldehyde. Similar, we have keto functional groups, and the class of compounds that contain these are ketones. And so it is a carbon double bonded to an oxygen and then also bonded to two groups that are not hydrogen. So an example of this would be this acetone molecule right here. And then we also have carboxyl groups. So those will come into play with amino acids. An example of this would just be the carboxylic acids. And so this is carbon double bonded to an oxygen and then bound to a hydroxyl group. So these are actually acids that are able to donate that proton in solution. And an example would be acetic acid. So it has this COOH group here. And if it loses that hydrogen, then it would be a COO group with a negative charge on it. Next, we have the amino functional groups, and the class of compounds that would contain these are amines. So these do obviously come into play with amino acids. And so the structural formula is NH2. They can also accept protons, which would make them NH3 positive, carrying a positive charge. And then an example of this would also be alanine or any other amino acid. So they have this amine group here, and then this carboxylic acid group here, which is where we get the name amino acid. Next, we have phosphate groups. And so organic phosphates and other types of molecules can be phosphorylated and carry this phosphate group. And so that's going to be a phosphate bound to 4 different oxygen atoms. and it carries a negative charge. And so an example of this would be 3-phosphoglyceric acid. It's got this phosphate group right here. And then the last group we're going to talk about are sulfhydryl groups. And so the class of compound that contains these are thiol molecules. And so this is a sulfur-containing group, so just SH shown here. And they can also lose or gain that proton. And so an example of this would be the amino acid cysteine. You can see it has this thiol group right here. All right, so now that we've talked about functional groups a little bit, we can get into the meat of the lecture, which is a discussion on amino acids. So amino acids are the building blocks of proteins. So proteins are just amino acid polymers. So they're just a string of amino acids that are folded in a particular way. So you can see on this figure over here on the left, we have individual amino acids. Those individual amino acids are going to be joined together with peptide bonds to form an amino acid sequence, a very specific sequence. And then that sequence is going to dictate how that polypeptide amino acid sequence folds into a three-dimensional protein structure. And so the amino acid sequence determines the protein structure. And then the structure of the protein determines its function. And so the individual amino acids on that protein are often referred to as residues. So I'll use that term throughout this lecture and other lectures as well. And so that structure of the protein actually determines its function, and proteins have a lot of different functions in your body. So some proteins catalyze chemical reactions, so those proteins would be referred to as enzymes. Proteins can also provide structural support to both your body and just to cells in general. Proteins are also used as cell signaling molecules, so they're ways that cells can communicate with each other and change the physiological state of different cells. Proteins are also involved in immunity, so antibodies are proteins. And they're also involved in generating motion. So if you think about your muscles, that is involving proteins doing that contractile activity, and also flagella in bacteria. So amino acids are the building blocks of proteins. So proteins are amino acid polymers. So all amino acids share a similar chemical structure. So all amino acids consist of a central carbon atom, which is shown right here. And that's connected to 4 different groups. So one is an amino group, which is shown right here. The other is a carboxylic acid group, which is shown right here. And also connected to a hydrogen right here. So that's where they get their name, an amino acid. So they have an amine group and a carboxylic acid group connected to that central carbon atom. And so this region right here with the amino group, the carboxylic acid group, and the hydrogen group are going to be conserved between different amino acids. So all different amino acids are going to have this central alpha carbon connected to an amino group, a carboxylic acid group, and a hydrogen. So how the amino acids differ is in this R group here, which is also called the side chain. So this R group is going to be variable and different between each of the different amino acids, and that's what's going to give them their unique chemical structure and unique chemical characteristics. Now, since the central alpha carbon on these amino acids is attached to 4 different groups, amino acids are going to be chiral, so they're going to have chirality. So they're going to form these left and right-handed mirror images of each other that are non-superimposable. Now, interestingly, only L isomers of amino acids are found in proteins on Earth. So the reason for this is unknown. So apparently, pure forms of L isomers or D isomers of amino acids are a bit more stable than mixtures. So you just have to think possibly, back in our evolutionary history, for some reason, the L isomer was the dominant form, and it's the only form of amino acids that are found in proteins on Earth. So we know all humans are made up of proteins made out of only L amino acids. But if there's some alien species or something out there that uses D amino acids, we've yet to discover that. But it's an interesting feature of amino acids. So all amino acids also contain at least two charged groups. So the amino group and the carboxylic acid group in amino acids can be protonated or deprotonated. So at physiological pH, so around pH 7, both the backbone groups are going to be charged. So that amino group shown here is going to be protonated and carry a positive charge, and the carboxylic acid group is going to be deprotonated and carry a negative charge at that physiological pH. So at pH 7. And so you just think back to our discussion on pKa's. So if the pH drops below, let's say at about 1, then that carboxylic acid group is actually going to become protonated, and the amino group will also be protonated. And then if we go up to, let's say, pH 12, both those groups are going to be deprotonated. So that amino group is going to be deprotonated, and the carboxylic acid group is going to be deprotonated. So this will change the net charge of the amino acid. So at pH 1, it's going to have a net charge of positive 1. At physiological pH, pH 7, it's going to have a 0 net charge because those positive and negative charges are going to cancel each other out. And at a higher pH, like pH 12, it's going to have a net negative charge because it just carries the negative charge from that carboxylic acid group. So look back at our discussion on pKa's, and this will make a lot more sense to you. But it's important to know that at physiological pH, that this is going to be the dominant form of amino acids. So they're going to have a positive amino group and a negative carboxylic acid group. And since both groups are charged, that the term for that is a zwitterion, so two ions. All right, so now that we've discussed some of the features that amino acids have in common, we can see how they are different. So there are 20 different amino acids found in proteins, and these can be categorized into 4 groups. So there are some amino acids that are hydrophobic. So they have non-polar R groups. And so 9 amino acids are going to fit into this category. So there's 9 amino acids that are hydrophobic with non-polar R groups. There's also going to be 6 amino acids that are polar, that have polar R groups, but that are neutral at pH 7. So 6 amino acids that are polar, have polar R groups, but are neutral at pH 7. There's also 3 amino acids that are positively charged at pH 7. So they have hydrophilic side chains, and those are positively charged at pH 7. And then lastly, there's 2 amino acids that are negatively charged at pH 7. So they have negatively charged R groups. So 20 different amino acids categorized into 4 different groups: hydrophobic, polar, positively charged, and negatively charged. So now we'll talk about the amino acids individually using those different categories. So we'll start out with the hydrophobic amino acids that have aliphatic R groups. So remember, aliphatic refers to those hydrocarbon chains. So there are 7 different amino acids containing hydrophobic aliphatic side chains. So the first one we'll talk about is glycine. So glycine, its R group is actually just another hydrogen atom. So it's the simplest amino acid. And it's the only achiral amino acid because it's the central alpha carbon isn't attached to 4 different groups. It's just attached to 2 different groups and then 2 hydrogens. So it's the only achiral amino acid. So I would like you to understand the structure of the different amino acids, their different characteristics, also their full name, their 3-letter code, and their 1-letter code that signifies that amino acid. So for glycine, the full name, of course, is glycine. The 3-letter code is GLY, gly, so that's easy. It's the first 3 letters in the name. And then the one-letter code is G, which is also easy. So G for glycine. So it's the simplest amino acid, and it just has a hydrogen as its R group. So next we have the second simplest amino acid, which is alanine. So alanine just has a methyl group as its R group. So it's the second simplest amino acid. So alanine is also often used in scanning alanine mutagenesis. So if you say you have a protein and there's a side chain at a specific position that, let's say, it's positively charged or something, and you think that has to, that's involved in its activity, you can mutate that residue to something simple like alanine that's not going to disrupt the structure of the whole protein, but will take away that functional group that you think has some influence on its activity. So that alanine is a very important amino acid for those types of studies. So alanine, it's the second simplest amino acid. It's alanine, ALA. That's also very easy. It's the first three letters in the name, and then its one-letter code is A. So now we come to the branched chain amino acids. So I'm going to talk about these three together because they have sort of a similar characteristic. So there's three amino acids that have these hydrophobic aliphatic branched chains for their R groups. So the first one is leucine. So leucine has this very bulky side chain made out of all connected methyl groups. So there's 1, 2, 3, 4 carbons on it, and it sort of forms this L here, which I think helps me remember the name leucine. So leucine, LEU, and then the one-letter code is L, so pretty easy. The next one is valine. So valine's similar to leucine, but it has only 3 carbons instead of 4 carbons. So 1, 2, 3. And I like to draw this sort of as a V. They have it drawn sort of in a L here as well, but I don't know. Hope maybe that'll help you if you think about it looking a bit like a V. So that's valine, VAL, and V. And then the last branch chain amino acid is isoleucine. So that is very similar to leucine in that it has 4 methyl groups that are branched, but the only difference is the position of this branch off the side of the chain here. So it's at this position, the beta carbon position here, where it's at, I think you'd call that the gamma carbon or something on leucine. So isoleucine, ILE, so it's not the first 3 letters in the name, and then the one-letter code is I. So those are the branched chain amino acids. So they're going to be very important for folding into the hydrophobic interior of a protein and sort of fit together like puzzle pieces with very, very little space. So these you can sort of group together when you're remembering them. So next we'll come to methionine. And so methionine has sort of an interesting structure. It has this long, unbranched aliphatic chain, but it has these 2 carbons and then a sulfur atom, and then another carbon here. So it actually has this thioether bond in the middle of the chain. So 2 methyl groups, a sulfur, so the thioether group, and then it ends in a terminal methyl group. So that's methionine. So you can think methyl and thiol. So methionine, 3-letter code MET, 1-letter code M. And then our last hydrophobic aliphatic amino acid will be proline. So proline has a pretty interesting structure. So it has this ring structure here. So its R group has 1, 2, 3 carbons, and then that terminal carbon is actually bonded through a covalent bond to the amino group. And so that's not found in any other amino acid. So it has this interesting ring structure. And proline often, for that reason, it's a bit more conformationally rigid than the other amino acids, and it actually breaks secondary structure in proteins sometimes. So that's proline, PRO, and then the one-letter code is P. So that one's pretty easy as well. So we can review this pretty quick and just some ways to sort of remember the different amino acids. So glycine just has a hydrogen as its R group, and it's the simplest amino acid, and it's the only one that's achiral. Alanine just has a methyl group, so it's the second simplest amino acid, alanine. Then we have our 3 branched chain amino acids. So we have leucine, valine, and isoleucine. So you can sort of remember those ones together. Then we have methionine, which literally it's a methyl group. So that, and it has sort of an interesting structure. So that should help remember it. And then we have proline, which has this interesting ring structure that you don't find in any of the other amino acids. So those are the 7 different amino acids containing hydrophobic aliphatic side chains. Okay, so remember that I actually said that there were 9 different hydrophobic amino acids. So we already met 7 of them that have hydrophobic aliphatic side chains. There's also 2 amino acids that have hydrophobic aromatic side chains, and they're phenylalanine and tryptophan. So phenylalanine is the simplest of the aromatic amino acids. So it's basically just alanine, but then one of the hydrogen atoms is replaced with this phenyl ring. So that should help you remember it. So it's literally phenylalanine. And phenylalanine is very important. It's a precursor for some neurotransmitters like dopamine as well that are used in cell signaling. So that's phenylalanine. The 3-letter code is PHE, so phe. So that's the first letter, 3 letters in the word, in the name, which is easy. But then its 1-letter code is F. So P is for proline. So we need a different letter for phenylalanine, and it's F. But that's not too hard because you can sort of remember phenylalanine. It sort of has that F sound. And then the other hydrophobic aromatic amino acid is tryptophan, which you might have heard before. So it also is sort of built off of alanine, but then it has this indole ring here. So it's the bulkiest amino acid because it has this large indole ring. So that's 2 aromatic rings that are fused together, and it has this NH group here. So it's the bulkiest of the amino acids. So it's also a precursor to some signaling molecules like melatonin and serotonin, and it's actually not found in turkey at higher concentrations than any other meat. So that's sort of I'm not sure how that rumor got started, but it is sort of bulky like a turkey. And if you think of this NH group as sort of the head and this phenyl group as a tail, maybe that'll help you remember the amino acids. So it's tryptophan. It's bulky like a turkey. Its 3-letter code is TRP, so not TRY, so tryptophan, 3-letter code. And then its 1-letter code is W, so it's not in the name at all. But if you think of W as kind of a bulky letter and tryptophan as a bulky amino acid, that also might help you remember. So we have 2 hydrophobic aromatic amino acids, so phenylalanine, so literally phenylalanine, and then tryptophan, which is the bulkiest amino acid. And you can remember it if you think of turkey, even though it's not present in turkey at higher concentrations than any other meat. But maybe that'll help you remember. So now we come to the polar neutral amino acids. So there's 6 different amino acids that contain a polar side chains that are neutral at pH 7. So the first one is serine. So serine is basically alanine, but with a hydroxyl group here on the terminus. And so it has this methyl group and this hydroxyl group. And serine can be phosphorylated. It's one of the three amino acids that have hydroxyl groups. They can have a phosphate group added to them that can change the activity of a protein. So serine has a hydroxyl group. Its 3-letter code is SER, which is easy, and its 1-letter code is S, which is also easy. We also have threonine, which is very similar to serine, except it also has this branch off the side of this extra methyl group here. So it's threonine. It can also be phosphorylated on its hydroxyl group. And I like to remember its name because it has sort of 3 different things. It has an OH group, a CH2 group, and then a CH3 or a CH group, and then a CH3 group. So threonine, 3-letter code is THR, and then its 1-letter code is T, which is also easy. And then we have the last amino acid with a hydroxyl group, and that's tyrosine. So tyrosine is our 3rd and last aromatic amino acid. So it looks just like phenylalanine, except it has this hydroxyl group here in the para position of that benzyl or phenyl ring. So tyrosine can also be phosphorylated at this hydroxyl group. And an interesting thing about tyrosine that I always like to think about is that it can actually snorkel in membranes. So if you think about this part of the molecule, that's a hydrophobic part of the molecule, whereas this hydroxyl group is a hydrophilic part of the molecule. So if it's embedded in a membrane, this hydrophobic group can actually be in the membrane, and this hydroxyl group can sort of stick out like a snorkel. So I enjoy snorkeling, so that helps me remember tyrosine. So it's tyrosine, TYR, and then its one letter code is Y. So if you just think of tyrosine, you can sort of remember that Y sound with it. And then we also have cysteine, which is very similar to serine, except it has this, instead of a hydroxyl group, it has a thiol group. So it has this methyl group here, and then this SH group at the end. So this sulfur group can actually be reduced and make disulfide bonds with other cysteine residues. And that's very important for protein structure. So that's cysteine, and I like to think of cysteine and serine together because they're sort of similar in their structure. So that's cysteine, CYS, and then its one letter code is C. And then last we have the two amide containing amino acids, which we can sort of discuss together. So they're asparagine and glutamine. So both of these amino acids have an amide group here on the terminus of their side chains. So that's a carbon double bonded to an oxygen and then bonded to an amino group. So that's called amide, an amide group. So and the only difference between them is how many of these methyl groups are on the chains before you get to that terminal amide group. So the first one is asparagine. So its three-letter code is ASN, and then its one-letter code is N. That's one of the harder ones, I think, to remember, but it's asparagine. It was actually the first amino acid that was ever isolated, and it was isolated from asparagus juice, which is how it gets its name. So it has one methyl group and then that terminal amide group. And then glutamine has two methyl groups before that terminal amide group. So glutamine, its three-letter code is GLN. And its one-letter code is Q, which is also kind of tricky, but I just kind of think glutamine, it's kind of a Q sound. I don't know if that'll help you. That's how I remember it. And I try and also a trick that I use to remember their names is that asparagine starts with an A, so that's the first letter in the alphabet. And then glutamine, that comes after A in the alphabet, so that's the second. And so asparagine is first, so it has one methyl group, and then glutamine is second, so it has two methyl groups. I don't know. That helps me. Maybe that'll help you as well. All right, so those are the 6 different amino acids that contain polar side chains that are neutral at pH 7. So I like to remember cysteine and serine sort of together because they're similar. Cysteine has a hydroxyl group. Serine has a thiol group. Threonine, I like to think of 3 things. There's the OH, a CH, and then a CH3. There's tyrosine as the snorkeler. It sort of looks like it's snorkeling, if that helps you remember it at all. And then there's asparagine and glutamine, which I sort of group together in my head. And asparagine comes first, so it has one methyl group before its amide group. And then glutamine comes second. The G comes second. So it has 2 methyl groups before its terminal amide group. So those are the polar neutral amino acids. So now we come to the positively charged amino acids. So there's 3 amino acids that contain hydrophilic positively charged side chains at or near pH 7, so physiological pH. So the first one is lysine. So lysine has this long aliphatic chain of 4 carbon molecules and then terminates in an amino group. And this amino group carries a positive charge at physiological pH. So that's lysine. It's a pretty interesting structure, and it actually shows you how dynamic amino acids can be within proteins because this region right here is actually hydrophobic. And then this region, the terminal amide group, is hydrophilic and positively charged at physiological pH. So that's lysine. Its three-letter code is LYS, which is easy, but then its one-letter code is K. So that's one that you're just kind of going to have to remember. And then similar to lysine, we have arginine. So you can sort of group these two together in your head. So it similarly has 3 aliphatic methyl groups and then a terminal guanidino group. Guanidino group here, and that carries a positive charge in its resonance structure. So it's 3 amino groups bonded to a central carbon, and it has resonance structure and carries a positive charge as well. And you can see it also has sort of this hydrophobic region and then a hydrophilic charged region at the end. And so it's about maybe the second bulkiest amino acid, or it's a pretty bulky amino acid, just like tryptophan. So it's arginine, ARG, and then its one-letter code is R, which is pretty easy if you just think arginine. And then we come to histidine. So histidine is a little bit interesting, and I'll talk about it on its own. So histidine has a methyl group, and then it has an imidazole ring as its R group. So imidazole group. And the interesting thing is that histidine can be positively charged at pH, at or near pH 7. So its pKa is sort of close to pH 7, so there's going to be a combination of positive and negatively charged uh, uh, histidine molecules, so that are either going to be protonated or deprotonated. So this is the protonated form, and this is the deprotonated form. And because it can gain or lose that proton, it's often very important in proteins that catalyze chemical reactions, so enzymes. So histidine's often involved in the active site of different enzymes, and for that reason, it's pretty important. But it can be positively charged at or near pH 7, so that's why we include it with the other amino acids. So it's histidine. Its 3-letter code is HIS, H-I-S, which is easy, and its 1-letter code is H, which is also easy. So those are the 3 hydrophilic positively charged amino acids. So there are lysine, and they're all pretty unique, so it's best to just remember them. So lysine has this amino group at the end of an aliphatic chain. Arginine has a guanidino group at the end of an aliphatic chain. And then histidine has an imidazole group at the end of its of just one group. So imidazole group basically coming off of what would be alanine. So 3 different positively charged amino acids: lysine, arginine, and histidine. So lastly, we come to the 2 amino acids that are hydrophilic and negatively charged at or near pH 7, and they're aspartate and glutamate. And so they're kind of similar to asparagine and glutamine, but instead of an amine group, they have a carboxylic acid or carboxyl group at the end of these chains. So it's very similar to asparagine and glutamine, so aspartate and glutamate. So I think of aspartate is A comes first, and it has 1 methyl group and then a carboxylic acid group here. And then G for glutamate comes second, and it has 2 of these groups before the carboxylic acid group. So they'll often be called aspartic acid or glutamic acid, but at pH 7, they're deprotonated and they carry a negative charge. So the correct term would be aspartate or glutamate when they're in this form. So there's aspartate, which is ASP, which is easy, ASP, first 3 letters in the name. But then the one-letter code is D, which is a little tricky, but I just think aspartate. I sort of make a soft T. And then the other one is glutamate, and that's GLUNE. And so if you've ever eaten ramen noodles or anything with MSG on it, so MSG stands for monosodium glutamate. And so this is the amino acid that you're eating that makes everything taste very yummy. So there's two hydrophilic, negatively charged amino acids, aspartate and glutamate, and I just use that naming sort of convention. A is first; it has one methyl group before the carboxyl. G is second; two methyl groups before the carboxyl group to help remember those two. Okay, so now that we've gone through all 20 of the amino acids and discussed their unique side chains or R groups, we can think a little bit about how those side chains are actually ionizable as well. So we talked about that with the amino group and the alpha carboxyl group, but there's actually 7 amino acids that have ionizable side chains as well. So we usually think about this as, at physiological pH 7 for each group, and its pKa. So we can actually go through this table and determine what would be the dominant form of the side chain at pH 7 for each ionizable group. So we already talked about the terminal alpha carboxyl group. So this is on every amino acid. So its pKa is 3.1. So if you remember, pKa would be the pH where both the acid and the base are going to be present in equal concentrations. So at pH 7, that's above the pKa of 3.1. So this base group, the negatively charged group, is going to be dominant at pH 7. Similarly, for the alpha amino group, which is shown right here. So its pKa is 8. So at physiological pH 7, the dominant group is going to be the protonated acid group, which is why it carries the positive charge. So we already discussed that. Now, if we look at aspartic acid and glutamic acid, they have these carboxyl or carboxylic acid side chains on them. And the pKa of that side chain carboxylic acid is going to be 4.1. So that's why this is going to be the dominant group at physiological pH 7, and that's why we refer to them as aspartate and glutamate. Now, histidine, we discussed, but its pKa is at 6, which is pretty close to physiological pH. So actually, both the positively charged and the neutral charged the protonated and the deprotonated form are going to be present at pH 7, although this deprotonated form is going to be the dominant form at pH 7. Now, going down the line, cysteine, the rest of them, basically the acid is going to be the dominant form. So cysteine has an ionizable side chain, so its pKa is 8.3, and this is going to be the dominant form at that pH. Lysine, its amino group, it has a pKa of 10.8, and so this is going to be the dominant form, the positively charged form. Tyrosine, it can lose that proton on its hydroxyl group, and its pH is going to be, or its pKa is at 10.9, so the protonated form is going to be the dominant form. And then lastly, arginine is going to be, its pKa of that terminal guanidino group is going to be 12.5. So this positively charged group is going to be the dominant form. But it is important to understand that the pH of a specific amino acid of the surroundings can be affected by the pH of the protein microenvironment. So if these amino acids are surrounded by other amino acids that affect the pH of the microenvironment, it can cause them to gain or lose these ionizable groups. And that's going to come into play a lot when we talk about enzyme activity. And if you actually look into the details of how reactions are catalyzed by enzymes, a lot of times it has to do with the gaining or losing of protons and charges in these ionizable side groups. And then the last topic I want to talk about is essential versus non-essential amino acids. So essential amino acids are those that are needed in our diet, whereas non-essential amino acids can be synthesized from precursors in our body without us having to ingest them. So here's a table listing non-essential to essential and essential amino acids. So we can use this opportunity to just quickly go through and review each of the amino acids with their 3-letter codes and their 1-letter codes. So first, the non-essential amino acids, we have alanine. So that's the second simplest amino acid. It just has that methyl group there, and it's ALA and A for its 1-letter code. Next, we have arginine. So it has that, it's positively charged at pH 7. It has that terminal guanidino group, and then its 3-letter code is ARG, and its 1-letter code is R. Asparagine, so it has that terminal amide group, so ASN and N. And then aspartate, which has the terminal carboxylic acid group, so it's negatively charged, so ASP and D. And cysteine, which has that thiol group, looks a lot like serine, so CYS and C. Glutamate also has that terminal carboxyl group, when it's negatively charged, so GLU and E. Glutamine, that's one of the amide-containing amino acids, so it's GLN and Q. Glycine, which is the simplest amino acid, the only one that's achiral, so GLY, G. Proline, which is hydrophobic but has that interesting ring structure, so that's PRO and P. Serine, which has the methyl group and the hydroxyl group, SER, S. Then we have tyrosine, the snorkeler, so it's like phenylalanine, but with a hydroxyl group on the end of the phenyl ring, TYR and Y. And so those are all the non-essential amino acids. And then we can move into the essential amino acids. So these are ones that we need in our diet. So histidine, which has that imidazole group and can be positively charged at pH 7, HIS, H. Isoleucine, which is one of the branched chain amino acids, ILE and I. Leucine, one of the other branched chain amino acids, LEU, L. And we have lysine, which is positively charged, LYS, K. Methionine, so it's like meth and thiol, has a thioester group, MET and M. We have phenylalanine, which is like alanine but with a phenyl group on it, so PHE and an F for phenylalanine. The threonine, which has 3 different things, similar to serine but with that methyl group, THR and T. And tryptophan, which is the bulkiest amino acid, so TRP and W. And then lastly, we have valine, so another one of those branched chain amino acids. So that's VAL, and then the 1-letter code is V. So those are the essential versus non-essential amino acids. All right, everybody, so that concludes lecture number 3, amino acids. And there are a few points that I want to summarize or emphasize at the end here. So one is that amino acids are the building blocks of proteins. So the specific order in which the amino acids are built are going to dictate the 3-dimensional structure of a protein and hence its function. You also need to understand the basic structure that all the amino acids share. So they have a positively charged amino group at pH 7 and a negatively charged carboxylic acid group in the backbone at physiological pH 7. So you need to understand that Zwitterion concept and how that can change with the different pHs. You also need to memorize the structures of each of the amino acids. So that's why I went through them so slowly and tried to give you tips on how to memorize the different characteristics of each of them. So you need to know the chemical structure, the full name, the 3-letter code, and the 1-letter code for each amino acid. So it's very important that you at least know how to reference that information, but if you know it off the top of your head, it's really going to help you with all the exams through this course because the amino acids are so important. And you need to definitely know the characteristics of each amino acid, whether it's hydrophobic, polar, or charged. You need to know that. So if I ask you what is a characteristic of glutamate, you need to know that it's negatively charged, lysine positively charged, valine hydrophobic, stuff like that. You should be able to sort of drill that into your brain so you know it for the rest of the course. So thank you very much. And if you have any questions, please let me know.